Good evening, dear friends. Yes, my voice is audible, Shweta. Yes, Shweta Padmavati.
Yes, sir.
Okay, what about your friends? So, shall I start class, dear friends? Very good evening. What are the last class we discussed? Yes, what are the last class we discussed? Yes, Padmavati Shweta, please respond. Yes, Padmavati. Okay. Yes. What about other members? Yes, please respond. Shweta. Okay. My voice is audible. Please respond. Anyone? Shweta.
Okay, sir.
Okay. My voice is audible.
Yes, sir.
Thank you. What about Padmavati? Padmavati. Padmavati. Okay. Shall I start class? So today we will discuss to the topic is fertilization. What is the fertilization? Unmute yourself, Shweta and who is she? Padmavati. Padmavati and Shweta, please unmute yourself.
Yeah, the disturbance of
Sir, okay, you can carry on. So, okay, no problem. So, fertilization. The fertilization means the fusion of male and female gametes during the sexual reproduction is called fertilization, right? What is the definition of fertilization? Fertilization in plants, the process of the or fusion of the male and female gametes during the sexual reproduction. It is called as fertilization. In this, in angiosperms, angiosperms, flowering plants. Male gametes are produced inside of the pollen, where it is formed in the male gametes. In angiospermic plants, the male gametes are produced inside of the pollen grains, and the female gametes inside of the embryo sac. Okay, clear? And there are various steps are involved in the procedure of the fertilization. After the process of fertilization in a flower, the development of zygote into a flower and further fruit take place. This is very simple. Already you studied in the previous classes in BSc level and intermediate level also. The process similar both angiosperms, which are the flowering plants, and gymnosperms, which are the fruit bearing plants also. Okay. So what is the detailed meaning of fertilization? The plant fertilization is the process of fusion of male and female reproductive cells or gametes to produce the fertilized egg, to produce a fertilized egg, which is a zygote. This process called fertilization. This zygote develops further and results in a flower, and after flower formation and later the formation of fruit in case of the gymnosperms. So the plants are primary producers, and plant reproduction is an important process as it helps production of various like food, cloth, oxygen. Like photosynthesis. The photosynthesis, what is the photosynthesis? Already I told you many times what is the definition. Considered as a so in this pollination, during the pollination, a pollen grains reach it the stigma. First of all, you see observe these are the flower fertilization diagram. What are the parts are there? The green part is the stigma and style. And below this, you observe this diagram: ovary, ovule, embryo sac. These are the downside is the sepals. It is protect from the uh, flower, and these are the filament. These are the anther, right? So this is the regarding about already I explained in the previous class. So here during the pollination, what happening in the pollination? During the pollination process, the pollen, the pollen is it is a male gamete is transferred to the female gamete through the some carriers. Result it's fertilization, right? Do you understand? Pollen is a process in which a pollen means it is nothing but the male gametophyte. Male gametophyte, male gamete is transferred to the female gamete through the some carriers. What happening in the finally? It results is the fertilization. These carriers can be either wind, either water, either insect, either animals. Okay. So the carriers is whether wind, water, or insects or animals. These pollens are generally present on the stigma. Where it is, these pollens are where it is located, it is generally present on the stigma of the plant. So the stigma is a part of the pistil, it is elongated part of the flower, and these pollens can be easily transferred to the female gametes for the fertilization. These are the pollen pollination during this process. Entire process is the during the pollination. So during the pollination, the pollen grains reach the stigma and germinate by the producing the pollen tube. The pollen tube grows down through the stigma and style and reach the embryo sac of the ovule in the ovary. Where it is located in the ovule, it is located in the ovary. Commonly, the pollen tube it enters the ovule through the microphyllae and discharges the male gametes into the embryo sac. One of the male gametes fuses with the egg and forms the formation of zygote. This fusion of male and female gametes is known as the fertilization. Or second, male gamete fuses with the two polar nuclei or secondary nucleus is the two have already fused in the previously and form of triple fusion nucleus called it is primary endosperm nucleus. The involvement of both sperms in the fertilization is called double fertilization. It is this double fertilization discovered by the Navashin in the 1898. Who is the scientist who discovered the double fertilization? Navashin, and the year is 1898. So these are the fertilization in the you observe that these are the diagram of pollen grains, anther, filament, and ovary, ovule, and petals. It is the embryo sac and sepals. This is the stigma, this is the style, this is the pollen tube, this is the sperm nuclei. How it is developed? The polar nuclei, it is egg nuclei, the formation of zygote, and the endosperm nucleus. It is the first of all the formation of endosperm, and after that seed coat, after that embryo, the seed. It is the seedling situation. These are the fertilization in the plant. So here. Uh, Coming to germination of the pollen grains. Germination of the pollen grains. Just you see this diagram. Once it is the longitudinal section of the gynoecium at the time of fertilization. Next, coming to how germinate the pollen grains. You know pollen grains? These are the pollen grains. Uh, Already we discussed in the during the pollination, the pollen grains reach the stigma and germinate by the producing the pollen tube. So the germination of pollen grains after reaching the pollen grains after reaching the stigma, the pollen grains begin to germinate. The stigma passes specialized surface for catching and holding of the pollen. The main function of the stigma to provide the pollen with water and necessary for the uh, germination. The stigma which secretes exudates are called wet stigmas. What we call wet stigmas. And this the plant example is Petunia, Steriligia, and Gia. And whereas the stigma which do not secrete exudates are dry stigmas, and the plant species Gossypium, Astraceae, Papaveraceae, Poaceae. So here the two types of stigmas are there. Some of the stigma which secrete exudates are called wet stigma, and some of the stigmas do not secrete any exudates. They are called dry stigmas. So this exudate is highly viscous and adhesive with a high reflective index and appear as a droplets on the surface of the stigma. So it is chemically, it consists lipids, sugars, amino acids, and phenolic compounds. Germination. This is the germination, and chemically it is consist sugars, lipids, amino acids, and phenolic compounds, right? So the germination of pollen grains, how pollen grains germinate, occur either immediately or it may take few hours to few days of their coming over to the stigma. It is depend upon the coming of the stigma. When the pollen grains reach to the stigma, then they germinate. This the germination is it is with immediately or few hours or few days, right? During the germination, the pollen grain is imbibes water, swells up, rupturing along with the germ pores. The intestine comes out from the long tube-like structure called pollen tube. What is this? Pollen tube. So here, this is the pollen grain. So here, this is the pollen tube. What we called the intestine comes out in the form of a long tube-like structure called the pollen tube. Normally, only one pollen tube is formed from each pollen grain. It is monosiphonous. It is listen carefully. Only one pollen tube is formed from each pollen grain. It is called monosiphonous. But in some Malvaceae, more than one pollen tube may be formed from each pollen grain. It is polysiphonous. Polysiphonous. Generally, only one pollen tube is formed from each pollen grain. It is monosiphonous. But some taxa like Campanulaceae, Cucurbitaceae, and Malvaceae, it is more than one pollen tube may be formed from each pollen grain. What we call this type? Polysiphonous. Polysiphonous. Siponus. So in Althea rosea is many 10 pollen tubes from a single pollen grains have been observed. What is the plant name? Is Althea rosea. 10, 10 pollen tubes. How many? 10 pollen tubes from the single pollen grain have been observed, and the Malva neglecta, the number goes up to it is 14 pollen tubes. As a rule, only one of them makes further growth and reaches the only. In this, out of 14 or out of 10, out of many, only finally only one it makes growth and reach the oval. Only one. The other one is degenerate in the different stages of the growth. So here, only one pollen tube reach to the oval. The remaining ones, they are degenerate in the different stages. The plant's pollen grains are grouped into pollinia. A large number of pollen grains may germinate in simultaneously to form several pollen tubes. Example, Asclepiadaceae and Orchidaceae. So the pollen tubes are branched in the Amentiferae, the branches which do not carry gametes, the functions as a asturia. So here I will share these are the types of styles. How it is styles are there. The first one is the styles is the type is A and B is the closed type styles, and CD it is open type closed. So here these are the regarding about germination of pollen grains. Okay, so how the pollen grains germination with the pollen grains, how to develop a pollen tube. These pollen tubes is monosiphonous, sometimes polysiphonous, sometimes the pollen tubes from many pollen tubes formed in the one pollen grains, and sometimes 10 pollen tubes from a single pollen grain, and some taxa, especially Malva neglecta, then the pollen tube number is 14. Generally, in this, whatever may be like 10, 20, 30, 40, but only here, only one pollen tube, it reaches to the oval. The other pollen tubes is degenerate in the different stages of the growth. So, the plants where pollen grains are grouped into pollinia. A large number of the pollinia means a pollen grains are grouped. Generally, we are previously discussed in the numbers, but here a group of, that means there is no countable. So group of pollen grains, what we call into pollinia. A large number of pollen grains may germinate uh, simultaneously to form several pollen tubes. Example, Asclepiadaceae and Orchidaceae. The pollen tubes are branched in Amentiferae, the branches which do not carry gametes have functions as asturia. So this is the types of two types of styles are there. One is closed, AB is the closed type, and CD is the open type styles. And next, growth and structure of pollen tube. How the growth, how to develop the growth and the structure of the pollen tube. Here the pollen tube grows between the stigmatic papillae and reaches the stylar part. The subsequent course of the tube depends upon the internal structure of the style. The style may be solid or hollow. These are the pollen tubes showing the Carol's plant. So here the style may be solid or hollow. Mostly the style is solid. Example is Brassica nicotiana arabidopsis. Since the growing tip of the tube separates hydrolyzing the enzymes, the enzyme is pectinase and make it with through the pectin reach all the layers. Pectin rich wall layer of the tissue. Uh, Contrary to this, Onianthera, Petunia, and Datura, the pollen tube grows through the intercellular spaces in the style. So in monocots, the styles are hollow and show a canal running continuously from the stigma to the base of the style. You observe this diagram. The stylar canal may be contained in the secretional fluid. Amaryllis and lilium may remain, it is dry, crocus. The secreted fluid contains a nutritive fluid for the growing pollen grains. When the pollen tube emerges through the germophore of pollen grain, the entire contents of the pollen grain move into the pollen tube. The entire contents of the pollen grains move into the pollen tube. The wall of the pollen tube is made up of the cellulose and pectin. The growth of the pollen tube is primarily restricted to its tip. The most of the cytoplasm is concentrated. The remaining part of the pollen tube and the pollen grain are occupied by the vacuoles. The cytoplasm is restricted to the tip of the pollen tube by callose plug. During the tube elongation, callose plugs are formed at regular intervals. These plugs divided the pollen tube into small segments. These plugs originate in the small rings, which grow centripetally, finally seal the tube. So, this is the pollen tube and callose plug. The tip of the pollen tube appears in the hemispherical transparent area. This is known as cap black. What we call it is known as cap black. The cytoplasm just behind the cap black, it shows numerous mitochondria, Golgi bodies, and endoplasmic reticulum and lipids. The Golgi vesicles are abundant and are rich in polysaccharides and RNA. So, the growth of pollen tube in the style always directed towards the ovary. Such unidirectional growth perhaps due to the hydrotropic and chemotrophic and mechanical factors. These are the reasons, the unidirectional growth due to the hydrotropic and chemotrophic and mechanical factors. The chemotropic substances leaching out the filiform apertures of the synergids attract to the pollen tube towards the embryo sac. The metabologically active substances like carbohydrates accumulate at the tip of the pollen tube, establishes the physiological polarity. So this also contributes unidirectional growth of the pollen tube. These are the way of unidirectional growth. In many taxa, the entry of pollen tube abdurata present in the placenta, funicules are integument germinate of the growth and germinate of the pollen tube. Next, formation of male gametophytes. How develop or how formation of the male gametophytes. So here, the male gametophytes are formed by mitotic divisions. male gametophytes are formed by the, what are the divisions? Mitotic divisions of the generative cell. Simply, what are the formation of male gametophytes? The male gametophytes are formed by the mitotic division of generative cell. Mitotic division of generative cell. The division usually from the anther. This is uh, uh, an example of Astraceae, Brassicaceae, Caryophyllaceae, and Poaceae. These are the example of family members. But in some species, the generative cell divides only after the pollen have been shed. These are the example is Rosaceae and Solanaceae family members. In those plants where the pollen are shed at two-cell stage, the generative cell may divide inside the pollen grain after it has settled on the stigma or in the pollen tube before it reaches the embryo sac. These are the formation of the male gametes. And next, how to entry of pollen tube into the ovule. How to entry? So you carefully observe this diagram. The entry of pollen tube into the ovule. Okay. So these are three types are there. Just wait. Actually, these are the three types. After reaching of the ovary, the pollen tube enters into one of the ovule and then into embryo sac. First of all, the pollen tube is after reaching of the ovary, the pollen tube first enter into the one of the ovule and then into the embryo sac. First of all, once it's reaching the reached the ovary, the pollen tube enters into one of the ovule, then into the embryo sac. This is the process of entry of pollen tube. Then the pollen tuber may enter the ovule through the one of the following three ways. This is the general way, but it is the pollen tube may enter in the ovule through the one of the following three ways. First one is the porogamy. Porogamy. What is the meaning of porogamy? Actually, these are the three types of entry of pollen tube into ovule. Porogamy, charajogamy, and mesogamy. The first one is these are the first one is a large number of plants. The pollen tube. You observe the diagram. Where is pollen tube? Where is microphyllae? The pollen tube enter to the ovule through the microphyll. Through the microphyll, this mode of entrance is called porogamy. Porogamy. So, what are the definition of this one? Porogamy. Simple answer. The pollen tube enters the ovule through the microphyllae. Very simple answer. The pollen tube enters the ovules through the microphyllae. This mode of entrance is called porogamy. This mode of entrance is called porogamy. Example is Ajadirecta, Oryza sativa, Nectanthis, Otilia. Okay. Entry of pollen tube in the ovule, into the ovule. How many times? 3 times. Porogamy. Very simple. The large number of plants, The pollen tube enters the ovule through the microphyllae. This mode of entrance is called as porogamy. What are the examples of porogamy? Azadirachta, Vorija sativa, Nyctanthis, and Otilia. And the second one is chalajogamy. Chalajogamy. In many amniferous taxa, the pollen tube enters the ovule through the chalajal end. Chalajal end. You observe the second diagram. Okay, you observe the this one. So chalajogamy means many amniferous taxa, the pollen tube enters the ovule through its chalajal end. Its chalajal ends. This is known as chalajogamy. Example is Casuarina. Another one is juglans. Another one is juglans. Next, the third one is mesogamy. Mesogamy. In this type of pollen tube enters in this type of pollen tube enters into the ovule through the microphyllae. Microphyllae. This mode of entrance is called in mesogamy, in this type of pollen tube enters into the ovule through the funiculus. Funiculus. It is this type of entry called as mesogamy, or the pollen tube enters through the integuments. It is called as mesogamy. So what is the mesogamy? In this type of the pollen tube enters into the ovule through the funicules. Example is Pisticia through the integuments. Example is cucurbita. These are the three types of pollen tubes enters to the ovules. First one is the porogamy, chalajogamy, mesogamy. So the porogamy. What is the meaning of porogamy? In many plants, large number of plants, the pollen tube enters to the ovule through the microphyllae. This mode of entrance is called porogamy. Example is Azadirachta, Vorija sativa, Nyctanthis, Otilia. Next one is chalajogamy. Next one is chalajogamy. The many amniferous taxa, the pollen tube enters to the ovule through the chalajal end. This is known as chalajogamy. Example is Casuarina juglans. And third one is the mesogamy. In this type of pollen tube enters into the ovule through the funicules. It is Pisticia, and through the integuments it is cucurbita. These are the three types of entry of pollen tube into the ovule. Next, coming to entry of pollen tube into the embryo sac. So in the previous one is the entry of pollen tube into the ovule. Now we will discuss entry of pollen tube into the embryo sac. Embryo sac. So it is irrespective of the place of entry into ovum. Irrespective of the place of entry into ovum, the pollen tube grows through the nucellular tissue and enters the embryo sac only at the micropolar end. The pollen tube may enter the embryo sac via one of the following routes we will discuss in. So this one, that is between the wall of the embryo sac and one of the synergids, between the egg cell and one of the synergids without destroying the latter, or directly into one of the synergids. You observe this diagram. This is the synergid cells, and before entry of pollen tube, degenerating synergids after the entry of pollen tube. These are the pollen tube. How it is the degenerating the synergy after entry of the, how it is degenerating. In the first one is the before entry of the pollen tube. This is the filiform aperture synergids pollen tube discharge. It is the some X bodies. These are the degenerating synergids. These are the vacuoles. So here the electron microscopic studies have shown the pollen tube enters into the one of the synergids through the filiform aperture. You know filiform apertures where it is located. Already we discussed in the previous class. As a rule, one of the synergid cell is destroyed even before the entry of pollen tube into the embryo sac. The rule of one of the synergid cell is destroyed and even before the entry of the pollen tube into the embryo sac. The other synergid cell survives and later disorganizes. This is the entry of pollen tube into the embryo sac. Next, we will discuss discharge of pollen tube contents. Already we discussed the pollen tube. It is consists of some contents. How it is discharged? The pollen the pollen tube grows through the filiform apertures. The pollen tube grows through the filiform apertures and enters the cytoplasm of synergid. At this stage, a pore develops either at the subterminal position, it is gossypium, subterminal position, we are seen in the example of gossypium, or the tip of the pollen tube. Example, Curcus and Petunia, both the sperms, male gametes, along with released into the synergid. These pollen tube cytoplasm do not mix with the cytoplasm of the synergid. The embryo sac where synergids are absent, the egg itself develops in the filiform aperture and receive the pollen tubes. It is seen in the plumbago capensis. These are the diagrams. In the below three diagrams are the sperm transfer into the embryo sac. How the sperm transfer into the embryo sac? The first one, the entry of pollen tube into the synergid, and B, the discharge of pollen tube contents, discharge of the pollen tube contents. And the third one is the proposed method of gametic discharge, the spermonucleus and X body. These are the sperm transfer of into the embryo sac or discharge of pollen tube contents. Next, fusion of gametes. How the gametes are fusion. So here, one of the male gametes approaches the egg cell, while the other sperm cells fuses with the plasma membrane, to the plasma membrane of the central cell. The membranes at the point of contact dissolve and the sperm nuclei are raised, one into the egg and other into the central cell. So the fusion of male gametes with the egg is known as syngamy. The fusion of the male gametes with the egg, what we call syngamy or true fertilization. Fusion of male gametes with the egg is known as syngamy or true fertilization. It results in the formation of a diploid zygote. So syngamy was first described by the scientist named Strasburger in 1884, who described in the syngamy. The syngamy described by the Strasburger in 1884. So the second male gamete fuses with the two haploid polar nuclei. The second male gamete fuses with the two haploid nuclei or diploid secondary nucleus, the forming of a triploid nucleus called primary endosperm nucleus. The second male gamete fuses with the two haploid polar nuclei, forming a triploid nucleus called primary endosperm nucleus. This fusion known as triple fusion. What we call this fusion. So what the fusion? Second male gamete fuses with the two haploid polar nuclei, forming a triploid nucleus called primary endosperm nucleus. This fusion is known as triple fusion. As the both male gametes are involved in the fertilization, the both male gametes are involved in the fertilization, it is known as double fertilization. What we called in double fertilization, the fusion is known as triple fusion, as both the male gametes are involved in the fertilization. It is known as double fertilization. This is a unique feature of the angiosperms and was first described with the Navash in 1898 in the Lilium and Fritillaria plant species. Lilium and Fritillaria. Next, the double fertilization is necessary for the production of viable seeds. The double fertilization is must and should. It is very, very important and very, very necessary because for the production of the viable seeds, it gives rise to endosperm, which is nutritive tissue for the zygote and embryo. The double fertilization, it gives rise to endosperm, which is nutritive tissue for the zygote and embryo. In angiosperms, the endosperm is post-fertilized tissue. So next one is X bodies. What are the X bodies? The synergid cell which has received the pollen tube. The synergid cell which has received the pollen tube discharge shows two darkly staining oval-shaped bodies. Two darkly staining oval-shaped bodies. Since the nature of these bodies was not clear, they were denoted as the X bodies. From the time to time, these X bodies have been variously interpreted. And uh, from the time to time of these X bodies have been variously interpreted. They are considered as remains of the vegetative nucleus, remains the synergic nucleus, cytoplasm of the sperms, and remains the degenerated megaspores, etc. Based on their shape, based on their shape and distribution in the cytoplasm, the presence of the DNA, it is reported by the Jensen in the 1972. It is interpreted one of them as the remains of synergid nucleus and other as the remains of the vegetative nucleus in the plumbago. And there are no synergids, only one X body is visible. It represents it remains a vegetative nucleus. So these are the regarding X bodies. And the next one is the polyspermy. Next one is the polyspermy. In many types, more than two sperms. Two sperms means two male gametes are released into a embryo sac. This condition is referred to as polyspermy. This may result because of the formation of more than one sperm in a pollen tube or due to entry of more than one pollen tube in the embryo sac. The polyspermy occurs only in the abnormality. Normally, an embryo sac receives only two sperms which take part in the double fertilization, but polyspermy may bring about the fertilization of the egg by one more than one male nucleus, or the supernumerary cells may fertilize other components of the embryo sac. These uh, synergids are these are the synergids are antipodals. This may result in the formation of more than one embryo in the embryo sac. So, if the embryo sac receives two or more pollen tubes, the sperm nucleus fusing with the egg nucleus may be derived for one pollen tube, and one fusing with the polar nuclei may be derived from another two. This condition known as heterofertilization. What we call heterofertilization. These are the regarding about fertilization. These are the fertilization. So just wait, we will go for the another one. And the what is the our syllabus? The next topic is sexual incompatibility. Next in sexual incompatibility. That is also we will cover. So here another topic is the sexual incompatibility in the flowering plants. So what are the reason of sexual incompatibility in the plants? So it is inability of certain otherwise viable gametes to fuse with each other and produce the fertile offsprings. The sexual incompatibility may be interspecific or intraspecific. The interspecific incompatibility is important. It prevents pre-cross pollination or pre-cross fertilization amongst the members of the different species. So it maintains the genetic individuality of the species. The intraspecific incompatibility is self-sterility or self-incompatibility is inability of the plant producing functional male and female gametes to produce fertile offspring when self-pollinated. So do you understand this one? It maintains the genetic individuality of the species, the intraspecific of incompatibility, the self-sterility or self-incompatibility is inability of the plant producing functional male and female gametes to produce fertile offspring when the self-pollinated. So it has a mechanism to prevent inbreeding and promote outbreeding. So self-incompatibility has been reported into 66 families of angiosperms. It is reported by the Me, Cuban, and Dickinson in the 1997. It can be due to the morphological or physiological reason. What are the reason of sexual incompatibility? The reason is due to the morphological and physiological regions. So what are the morphological self-incompatibility? So the morphological, there are 2 or 3 different matting types. It is distally, it is primula, and tristally, lythrum. These are the some morphological self-incompatibility of the flowering plants. So what is the physiological self-incompatibility? Physiological self-incompatibilities. There is no morphological distinction amongst the different matting types found in the species. The physiological self-incompatibility is 2 types. Again, this one is the 2 types. One is the gametophytic self-incompatibility, GSI, and another one is the sporophytic self-incompatibility, SSI and GSI. So gametophytic self-incompatibility, the incompatibility is due to genotype of the pollen. Example is Liliaceae, Solanaceae, Poaceae. These are the 3 family members or examples of the gametophytic self-incompatibility. The of pollen. For the reason, genotype of the pollen. Example is Liliaceae, Solanaceae, and Poaceae. It prevents pollen germination. Just wait. Okay. So it prevents pollen germination, causes retardation of the pollen growth, deterioration of pollen tubes, and failure of the nuclear fusion. So another one is the sporophytic self-incompatibility, SSI. So the incompatibility is due to genotype of the sporophytic or stigmatic tissues. The example is Ostraceae and Brassicaceae. The genetic basis of the self-compatibility was produced by East and Mangelsdorff in the 1925 in the form of multi-allelic single gene It is S gene. It contains over 30 alleles in Brassica oleracea. The pollen grain contains a single allele, while the stigma has two alleles. In the pollen grain, allele is similar to any of the two allele of the female parent. Say S1, S4, the incapability will ensue. So, some importance of the self incapability prevents with the self-pollination. So these are the some points they are given in the importance. Self-incapability preventing the self-pollination. The second one is the self-incapability has made the plants outbreeders of the outbreedings maintains the vigor and vitality of the race. The variation appears due to the outbreeding that provide the adaptability to changes in the environment. And the self incapability has defect. It does not allow raising pure lines required for the breeding programs. These are the some important points of the sexual incapability. So again, we will so once again it is you see the flower diagram. The hermaphrodite bisexual flower with compatible pollen showing germination to affect fertilization. These are the pollen grain, these are the pollen tube, the anther, that is the filament, the total is stamen, that is the petal, the ovals, is the sepal, is the ovary, the yellow color is the pollen grain, the upper side, these are the pollen tube, this one is the stigma, this one is the style, and ovary. This entire part is the pistil. This one is the stamen. So, journey from microspore to pollen grain. How develop, already we discussed in the previously, how develop in the microspore to pollen grain. Then we will discuss in the sexual incapability. Well equipped functional pistil when pollinated with viable functional pollen grains fail to form silk. It can be seen between the individuals of same species. It is the self-incompletely and different species. The classification of self-incompletely, this is types heteromorphic self-incompletely and homomorphic self-incompletely. So these are the disty, tristy, sporophytic self-incompletely, gametophytic self-incompletely. So you observe that these two different plants. It is diskly, tristly. It is primula plant having two kinds of flowers. It is controlled by one gene, two alleles. Here in the right side, the lythrum having three kinds of flowers controlled by two genes with two alleles. In the distally, it is controlled by the one gene, two alleles. Here, two genes and with two alleles. So these are the diagram. So you show this is the gametophytic self-incompatibility, sporophytic self-incompatibility, type pollination donor, genotype pollen, stigma. This is the entire stigma and style. This is the ovary, is the oval. And these are the difference between the gametophytic self-incompatibility and sporophytic self-incompatibility. GSA1, gametophytic self-incompatibility controlled by the genotype of the pollen grain. The control by the genotype of the saprophytic tissue is the plant from which is pollen is derived. So, anther. It is commonly tetrasporangiate containing the four sporangium. The number of cells in this sporangium act as a microspore mother cell ready to undergo meiosis from the microspores. Already we discussed in the previous class. They are initially held in the tetrad with callus. Upon enzymatic degradation of they become the free as a single microscopic having the thin wall of callus. After this development of pollen, a third pollen wall exam starts to protect the pollen grains, so they complete their journey. The female reproductive organs successfully. These are the microspore and prime exit between the plasma membrane and callus wall. Deposition of the sporopollenin at the distinct position in the primate cell. The PM starts separation from the callus wall, forming the undulation mark in the appears. There is no need of for reverse. So here so coming to today class. So what about fertilization? What is the definition of fertilization? Fertilization. Yes. Yes, are you there? Please respond once you recollect. What is the fertilization? The fusion of male and female gametes. Fusion of male and female gametes during the sexual reproduction is called fertilization, right? In angiosperms, the male gametes are produced. Where it is the produced in the gametes, the male gametes are produced in the pollen grains. The female gametes, it is also called as the egg cells inside the embryo sac. Inside the embryo sac. So here, so just you see the observe the diagram. So when will happens in this fertilization? In this fertilization during the pollination, the pollen grains reach the stigma and germinate by the producing the pollen tube. The pollen tube grows down through the stigma and style and reach the embryo sac of the ovule in the ovary. Commonly, the pollen tube enters to the ovule through the microphyllae and discharges the male gametes into the embryo sac. This one of the male gametes fuses with the egg and forms the zygote. This fusion of male and female gametes is known as fertilization, right? This is the
Yes, sir.
fertilization. Very simple already. So, but some of the important major points: how germination of the pollen grains, how growth and structure of pollen grains, and also uh, the pollen tube, structure. What are the how the formation of gametes like sperm cells, the vegetative nucleus, callus, plug, pollen tube. These are the very important, and also formation of male gametes. How the male gametes are formation, and how to entry of pollen tube into ovules. This is also very important in exams. They are asking the yes, entry of pollen tube into the ovule. So, of the pollen tube after reaching the ovary, the pollen tube enters into the one of the ovules, then into the embryo sac. First of all, the pollen tube enter to the ovule
Oval.
ovary. The pollen tube enter into one of the ovules. First ovary, then ovary to ovules, and then embryo sac. Embryo sac. So the pollen tube may enter the ovules through the one of the following three ways. What is this? What is the three ways?
Porogamy,
Chalagamy,
chalagamy, mesogamy.
Very simple. What is the porogamy?
Yes.
What is the definition of porogamy?
The pollen tube enters the oval
Yes,
through the microfile.
right. This mode of entrance is called as the porogamy. What is the example of this porogamy? Aja
Aj-
directa,
uh,
Varaiza
Arise
sativa,
sativa, nicto,
Nictanthis, Patilia. These are the examples of porogamy. Next, chalajogamy. So the answer is in the question only. Chalajogamy.
Enters
In,
through the chalazal
the
in,
pollen
in,
tube,
in,
the
in
oval
and
through its chalazal end.
calendar.
okay, but here one only in many Amentiferous taxa or Amentiferous genus, Amentiferous species. Only in especially in this Chalazogamies, Amentiferous plants, the pollen tube enters in oval through the chalazal end, right? So this is known as Chalajogamy. What are the examples of Chalajogamy?
Casuarina.
Casuarina and Juglans. Juglans. Next, Mesogamy. Meso. So here two ways are there. In this type of pollen tube enters into the oval, through the funicles
Through the
or integuments. What are the examples of funicles? Pistacia and integuments. The example is cucurbitaceae. These are the examples of the mesogamy. These are the very, very important. And also entry of pollen tube into the embryo sac. Before that, the entry of pollen into the oval, right? So here, entry of pollen tube in the embryo sac. So this is also already given 3 points between the wall of the embryo sac and one of the synergids. First one is between the wall of the embryo sac and one of the synergids. The second one is the between the egg cell and one of the synergids without destroying or directly into one of the synergids. These are the entry of pollen tube into the embryo sac, right? So these are the few very, very important of this regarding this one. So next class we will discuss what are the remaining units are there. We have book. So fertilization and sexual incompatibility. Okay. So embryology of angiosperms 2. So it is 4 chapters are there, 4 units are there. First one is the development of endosperm and development of embryo and apomixis and polyembryonic. So here the next class we will discuss the development of endosperm. What is the endosperm? Any idea?
Endosperm layer.
Nuclear, cellular, and no.
Yes. Anyone? Endosperm, embryo and endosperm is both are the two different. Okay. Endosperm. The endosperm is the nutritive tissue for the developing embryo, phanerogams, and gymnosperms. So in phanerogams, what is the phanerogams?
Non-flowering.
So what happening in the endosperm in this phanerograms? Endosperm is the nutritive tissue for the developing embryo. So before developing embryo, the embryo needs some nutrition. So the endosperms provide the nutrients to the developing embryo, right? In the phanerograms. In the gymnosperms, the endosperm is haploid. Haploid means
Single set of
is one set of chromosomes and is formed before fertilization. The gymnosperms, the endosperm, the formation of endosperm in the gymnosperms, it is before fertilization. So in angiosperms, it is triploid. Triploid means 3N, 3 sets of chromosomes, 3N, and developed from primary endosperm nucleus here after fertilization. After fertilization. So you observe the difference between the endosperms is the nutritive tissue. All the in gymnosperms and angiosperms in the phanerograms, the endosperm is it is a nutritive tissue, right? Nutritive tissue. So the nutritive tissue for the developing embryo in the phanerograms. In the gymnosperms, the endosperm is the nature is it is applied. It is applied in the one member, 1N, and is formed before fertilization. In the angiosperms, after fertilization. So in angiosperms, the endosperm is the triploid. Triploid means how many numbers? 3N. 3N. And developed from what is the first one? Developed, it is the endosperm nucleus. Endosperm nucleus. Primary endosperm nucleus. It is the after fertilization. In generally the angiosperm members, it is triploid in the 3N condition, but some number of polar nuclei varieties and different types of embryo sacs, the cytological nature of endosperms so also shows in the some variation. For example, Onenthera type of embryo sac, there is only one polar nucleus. The endosperm is diploid, 2N nature and 2N condition, and the Peperomia type of embryo sac has 8 polar nuclei. Hence, the endosperm is 9N member. These are the endosperms. We will discuss in the next class. Endosperm. What are the endosperm? How it is developed the endosperm and the types of endosperms. So the types of endosperms also in the nuclear endosperm, cellular endosperm, halobial endosperms. In the 3 times are there. So here another endosperms is the ruminate endosperms are there. So alluvium layer is there. So these are the covering the endosperm. Then after the endosperms, endosperm development, we'll discuss is the embryo development. Embryo development. Then again we will discuss what is the zygote, what is the embryogenesis, what is the dicotyledonous embryo, what is the monocotyledonous embryo, how development of embryo and the different types of embryos are there. So how and also we will compare with the monocot embryo. So once the embryonal adjustment is completed, then next one is embryology of the angiosperms, like parthenocarpy, experimental embryology, applications of embryology, taxonomy, agriculture and horticulture sectors. Principles of applications of the palynology is the last of our syllabus. So here How many classes remaining?
2 చాలా ఉన్నాయి దీనికి సగం సగం
27th is the last class. The practical schedule are released or not?
12 14 16 18
21st
20 22 24 26
27
7 classes.
It is 29th.
27 is last class.
Okay. I think before that I will finish the syllabus. What about assignment? Kumar sir are given to the assignments or not?
Yes, sir, given, sir.
What about this paper? Paper 6, medicinal botany and embryology paper. This is also sir given to this paper also, paper 6 also. Yes.
Sir, wait, sir, seeing. Given, sir.
Okay. You know how to write the you got some instructions how to write assignments, right?
Yes, sir, last year we wrote, no?
Yes. So any topics you write neatly will get the full marks. Well labeling, well draw the diagrams, and already I discussed in the previous classes also. You draw the diagram very neatly and the label mostly in the right place. In case the too much of labels is there, you are using the left space and right space also. Then you will write the assignment neatly, step by step. Okay, so you will get good marks.
Okay, sir.
So what about the practicals? You don't know?
Don't know, sir.
Okay, I think after the completion of these online classes, most probably the practicals. After practical, you go for the examinations, right? These are the, I think, plan. I'll take the your exams. I think practical exams April or May. April last week or May. So total 16 days, right? In each paper only 4 days, na?
Yes.
Yes, sir.
So that's the. So what about to other members? Daily only 4-5 members are there. 1, 2, 3, 4, 5. So what about others?
Don't know, sir.
Just attend the class, it's better. Otherwise, they are lose classes. Okay. And one thing, you are medicinal botany students, na? So you got the previous like material medicinal botany cultivation, ethnobotany, pharmacognosy. What about these notes? Do you have notes in the previous classes?
We don't have, sir.
You attend the online offline class, right?
Only 2 classes I have attended, sir.
Okay. So once complete all the syllabus, just you let me know what are the notes are required to you. Please send me not now, after entire syllabus. Then I will provide some notes. So
Okay.
remember, in case there is not there is some topics not available with me, you choose internet, you choose some books, you choose in the other ways also. Okay.
Okay, sir.
So that's better. Okay.
Okay.
Okay, next, any doubts regarding this class?
No, sir.
Yes. Soumya, Saraswati, Sasikirtana, Prashanti, Padmavati. Only 1, 2, 3, 4, 5. Only 5 members are there. Okay. Every day I took the attendance also. So just you inform your friends. This is a good time for you, no? The evening time. So after 7:30, one more class is there.
Yes, sir. 8 o'clock.
Oh, 8 o'clock. 8 to 9. That's
9:30, I think.
8 to 9:30. Who taken that class?
Kumar sir
Kumar sir. Okay.
and Ramakrishna sir. Other day Ramakrishna sir took the class and
Okay.
next Kumar sir.
Next Kumar sir. Okay. Okay. Thank you.
Okay, sir.
Thank you.
Thank you. Sir.
Thank you.